Welcome back to my channel Xgene and proteinX. In this tutorial video, I'm going to talk about the CRISPR-Cas9 mediated gene editing. In my previous two videos, I already discussed about the CRISPR-Cas classification and the mechanism and molecular mechanism. But before going to the main discussion, I would like to have some quick recap of this CRISPR-Cas9 mechanism. So CRISPR-Cas9 mechanism, it has two different phases, acquisition and the interference phase. In the acquisition phase, when the virus and the phage virus, when it attack the bacteria, then it incorporates its genome into the bacterial cell, and then the bacteria it has the CRISPR-Cas system. So by this system, the specific segment of the DNA sequence is copied and then it assembled in the CRISPR locus, and that segment of the sequence is called the spacer sequence. So in the CRISPR locus content spacer sequence, some repeat sequence, and Cas genes. Cas genes is actually the nucleus proteins that basically cleave the foreign DNA by recognizing some specific sequence that is known as the PAM sequence. And in the next phase, that is the interference phase, so that CRISPR locus that processed for the precursor CRISPR RNA, this is a transcription, and then the precursor CRISPR RNA it converted to some individual segment of the CRISPR RNA by RNase 3 enzyme. So that segment of that small segment of the CRISPR RNA it associates with the Cas9 proteins, and then when the phage virus when second time attack and it contains the same genome, so that genome contains the sequence that match with the CRISPR RNA, and then CRISPR RNA it has the complementary sequence of that genome of the viral DNA, and then it bind with that viral DNA. So that CRISPR RNA that is called the guide RNA, and then the Cas9 it recognize that DNA RNA duplex, and then it cleave the foreign DNA in certain portion that is known as a PAM sequence. So let's talk about the gene editing. So in the gene editing, there are two different proteins that are very much used widely. Those proteins are Cas9 and dCas9. So we already know about the Cas9, that is the nucleus enzyme, nucleus proteins that the main role is to just cleavage the foreign DNA or target DNA. But the dCas9 is the modified version of the Cas9. So basically the dCas9 it has some 2-point mutation. So this is the modified version, and this dCas9, because of this point mutation, it won't cleave the target DNA, it just regulates the transcription of the gene. So this is the dCas9 act as a repressor or the activator of the genes. Okay, let's talk about the CRISPR-Cas systems. So CRISPR-Cas9 system does not make cuts in the genetic sequence, and it does not permanently inactivate genes because it contains two different point mutation. So basically this CRISPR-Cas actually it has no endonuclease activity. Alright. So when the target DNA contains some part of the sequence, and that sequence match with the guide RNA, and that guide RNA bind with the target DNA, then the CRISPR-Cas system it bind with the promoter, and then it regulates the gene transcription by activating or suppressing. So eventually the dCas9 it won't cleave the target DNA. So then the enhancement or reduction of the transcription of the gene occurs. So corresponding proteins expression it regulated by this way. So in this CRISPR-Cas9 system also it contains multiple guide RNA can be designed at the same time, and that guide RNA it has some complementary sequence with the target DNA, and then when the guide RNA it bind with the target DNA, then the DCAS9 system it recognize that DNA RNA duplex, and then DCAS9 it regulate the transcription of that gene by binding with the promoter. Alright, so next I wanna show you that what is the difference between the DCAS9 and Cas9 structurally. So I aligned the amino acid sequence of this Cas9 and DCAS9 by MegaX software. So the alignment I am not showing here, but this is the alignment result. So as you see in this figure, in this schematic diagram, the Cas9 is the lower one, this lower one amino acid sequence, and the DCAS9 is the upper one. So as you see, all of the parts of the amino acid residues are same, except there are two different mutations. So in this portion, that is the 10 position. So in this 10 position, the D is mutated to 10. So actually the aspartic acid to alanine. And the another mutation that is in the position of the 840. So in this 840, the histidine it mutated to alanine. So this 2-point mutation it occurred in DCAS9 system. So the DCAS9 because of this mutation, DCAS9 it lose its cleavage activity or endonuclease activity. So that's why with DCAS9 we use just for suppressing or just for activating the gene transcription. So here is the plasmid that normally people they design this kind of plasmid for the CRISPR-Cas9 gene editing. So what inside in this plasmid, it contains Cas9, that is the main Cas9 that cleave any portion of the gene, and it contains also the guide RNA. This guide RNA contains some 1 to 20 nucleotide long sequence that match with the target DNA. So as well as also it contains U6 promoter. So this U6 promoter it actually promote the guide RNA, and also there are also some other sequence like ampicillin resistance genes, EGFR, but the main portion are the U6 promoter, guide RNA, and the Cas9. So this is basically corresponding to the CRISPR-Cas9 system. And for the DCAS9 system, everything is same. Only in place of the Cas9, the DCAS9 will be replaced. That's the difference. Alright, so let's talk about the application of this CRISPR DCAS9 system. So CRISPR DCAS9 system it based on artificial transcription factors. So but before going to the discussion of the artificial transcription factors, we have to know about what is the artificial transcription factor. Actually, the artificial transcription factors are used to express or suppress target genes. So actually this consists of the artificial transcription factors consist of the molecular domains such as the DNA binding domains that confer sequence specificity and may target similar sites in the genome with different affinity degree. So in the DNA binding domain, it bind some part of the selected DNA, and then it edit change the gene expression. So basically there are three different kind of this kind of domain, like DNA binding domain. The first one is the gene fingers domain. So this is also used for the gene editing as well as the transcription activator like effectors. This is another kind of gene editing system it used. This is another kind of DNA binding domain, and the most important one is the CRISPR-Cas9 system. So CRISPR-Cas9 system is also used for the gene editing. We well know that. So actually CRISPR-Cas9 contain two different kind of domain used normally. So usually for the gene repression, CRAV domain used only for dedicatedly for the gene repression in case of the gene repression, And in case of the gene activation, where we need to enhance the transcription, so in those cases we need to use three different kind of the domain, that is PP64, P65 and RTA. These three different kind of the domains associated with used combined this domain it used in case of the gene activation in the CRISPR-Cas9 system. So let's talk about the application of the CRISPR-Cas9. So there are this kind of CRISPR-Cas9 it widely used these days, and in the clinical trial there are a lot of trial is ongoing and many trials already done. So let's talk about that. So we know that the digidoxin and the chlorpromazine did already in the market drug. Those are used for the cardiac glycosid and antipsychotics, but it has some very mild effect on cancer, like expression of the P21 and other kind of effects. But when associated with it used with some complementary gene regulation with CRISPR-Cas9, then it has been found that hypoxia-inducible factor 1 and 2 it suppressed, as well as P53 it expressed. So these are used for different kind of the cancer. Another application is there are three different kind of the drugs: ritonavir, nelfinavir, and naproxen. These drugs are used normally for antiviral and NSAIDs, and these drugs also it has some lower effect on cancer. There are some cancer marker it expressed by this kind of drugs, although these kind of drugs are not used as an anti-cancer drug. But when these kind of drugs are used associated with CRISPR-Cas9 system, then it has been found that there are some significant effect that on expression of the P21 and as well as PI3K, this kind of regulation occurred. And eventually these kind of drugs are used with CRISPR-Cas9 system. It has been associated and the combined application is used for different kind of the cancer. Another kind of application is the ibuprofen and aspirin. We know very well that these drugs are anti-inflammatory drugs, and these drugs also has some very mild effect on cancer. Some kind of markers are regulated by these kind of drugs, but those kind of drugs are also not used as well as anti-cancer drugs. But when complementary gene regulation with CRISPR-Cas9, then it has been found that some kind of gene expression it was significant, like COX enzymes, and that's why this kind of drugs and combination with complementary CRISPR-Cas9 system is used in different kind of the cancer. So another kind of application is metformin and artesunate. So one is the anti-diabetic and another one is a microbiological agent. So both of these drugs, these are not the anti-cancer drugs, but those drugs having some mild effect on cancer as well as previous. So this artesunate it has some effect on some apoptosis marker like caspase-3, but when those drugs are used with CRISPR-Cas9 system, and then it has been found that some suppression of the anti-apoptotic proteins it is significantly, and also some MYC oncogenes. So we know that MYC oncogenes is very much widely known for initiation of the cancer. So when this CRISPR-Cas9 used with these drugs, then MYC oncogene it suppressed. So this system also used for different kind of the cancer. So those are CRISPR-Cas9. So let's talk about the CRISPR-Cas9. Also there are different kind of the application of the CRISPR-Cas9, and out of that beta thalassemia, this is most important use of this CRISPR-Cas9. And we know that HBB, these genes are poorly regulated, is dysregulated in beta thalassemia. Actually it's not very high expression in the beta thalassemia, but when CRISPR-Cas9 used, and then it has been found that it restored the HBB gene expression, and eventually beta thalassemia it recovered. So another kind of application for the CRISPR-Cas9 is the sickle cell anemia. So in this case, this is also for the HBB gene is not regulated very well. So it has been found when used with the CRISPR-Cas9 that this kind of is sickle cell anemia, it recovered. So another kind of application is the hemophilia A. This is very much important these days because hemophilia A, it actually it initiates because of the deficiency of the factor 8. So when used the CRISPR-Cas9 system, then it has been found that the factor 8 is expression is restored, as well as the hemophilia A regulated well. So another kind of the application is the hemophilia B. So hemophilia B is mainly actually the outcome of factor 9 deficiency. So when CRISPR-Cas9 used, also here the factor 9 is restored. Alright. So those all are very much important application, as well as there are a lot of applications these days. So another kind of application I would like to mention here. This is very much rare application. This is very much actually complicated kind of disease, and in those cases CRISPR-Cas9 used directly into the retina. So this kind of disease, you know that this kind of disease is not very well known, but this is one of the hereditary disease, and this is the LCA, liver congenital amaurosis, and for this kind of disease the childhood blindness occurred. So basically this kind of disease actually it occurred because some essential proteins that is very much crucial for cone and retina, and when there are some point mutation found in that genes, then that protein is not very well regulated and not well produced in the body. So for that this kind of disease occurs. So what happened then, we use CRISPR-Cas9 to mediate that kind of gene editing and to restore that kind of proteins expression. So we do normally the scientists, they did actually in the DNA, and that the DNA is packaged with inactivated virus, as you see in this figure inside the DNA, and it is packaged with the inactivated virus, and that virus it carry this DNA to straight to the retina, and then we injected this package directly to the retina, and that package contain also the CRISPR complex. Alright, so basically here is not directly the retina, it is in the eyeball. Actually is directly inject in the eyeball. And then what happened, the protein that I was talking about, that is CP290, this protein is very much essential for the cone and rod cell function. So when there are some point mutation occurred, then this protein's production actually it's suppressed. So basically for that reason this disease occurs, and the outcome is very much complicated, that is the childhood blindness. This is one of the genetic disorder. But when this kind of CRISPR-Cas9 it injected, it has been found that CP290, the mutated gene is edited and modified, and also this kind of disease it could be recovered. So another kind of application, that is the how ex vivo and in vivo genome editing is executed in clinical, that I'm gonna show here. So what happens here, so first the somatic cells are collected from the patients, and somatic cells and also the hematopoietic stem cells. So those kind of cells are collected from the patients, and these kind of cells are reprogrammed with CRISPR-Cas9, and the CRISPR-Cas9 contain the different kind of the system, like gene addition or gene deletion or the gene correction. So it contains different kind of the systems, and then it reprogrammed and the reprogrammed cells known as the iPSC. So iPSC actually basically the induced pluripotent stem cells, and this is a reprogrammed cells, and these cells are used for therapeutic purposes. And the next step, when this kind of these cells are reprogrammed, and then these cells are injected to the mice for evaluation purposes, and then eventually those reprogrammed cells directly inject to the patients. So this is how ex vivo and in vivo genome editing it execute. So this is all about the CRISPR-Cas9 mediated gene editing. Also, there are a lot of applications, so I will make another kind of video that what kind of recent application is ongoing. So I'll make another kind of video for that. So I hope this video will be helpful. If you like this video, kindly hit the like button, share it, and please subscribe my channel. And if you have any queries, kindly write in the comment section. Thanks.